podcast number 2, our second in a series of podcasts that will illuminate the workings of the nervous system. Before we begin, let's do a quick review of what we covered in last week's podcast. We began by learning about the various cells of the nervous system. We defined the functional unit of the nervous system as neurons. These cells have a soma, tree-like dendrites, and a single axon extending from the soma that carries the electrical signal all the way to the axon terminals. The enlarged end of the terminals contain vesicles filled with neurotransmitter molecules that are released into the synapse between the cells when the action potential arrives. The second type of cell in the nervous system is the glial cell, of which there are 4 types: Schwann cells, oligodendroglia, astrocytes, and microglia. Schwann cells and oligodendroglia are responsible for producing the myelin sheath on peripheral and central nervous system neurons, respectively. Astrocytes regulate the extracellular environment of the neurons and provide physical support and nutritional assistance. Microglia, on the other hand, act as phagocytes to remove cellular debris and provide immune function. Now, let's move on to today's lesson, resting membrane potential. So, what is resting membrane potential? Before we can begin to answer this question, let's take a step back and look at the bigger picture before zeroing in on resting membrane potential. The transmission of information within a single neuron is an electrical process and depends on the semi-permeable nature of the cell membrane. When the normal resting electrical charge of a neuron is disturbed sufficiently by incoming signals from other cells, a threshold is reached that initiates the electrical signal, the action potential, which then conveys the message along the entire length of the axon to the axon terminal. So, as we begin our research into the nervous system, remember that there are 4 important stages in the electrical transmission within a neuron: the resting membrane potential, local potentials, threshold, and action potentials. Now, let's zero in on the resting membrane potential. All neurons have a difference in electrical charge inside the cell compared to outside the cell. This is called the resting membrane potential. It can be measured by placing an electrode on the exterior of the cell in the extracellular fluid and placing a second electrode into the intracellular fluid inside the cell. This image demonstrates the first experiment that determined this characteristic of neurons. Researchers used a giant axon from a squid to take recordings of the resting membrane potential, as represented in these 2 images. The top image shows that when both electrodes are applied to the outside of the membrane, no difference in potential is recorded. However, when the microelectrode is inserted into the axoplasm, a voltage difference between the inside and outside is recorded at -70 millivolts. Therefore, the neuron is polarized in its resting state, meaning it is in a negative resting state. So, this is the first important characteristic to remember about the resting membrane potential, that the neuron is polarized in its resting state. But how does this resting membrane occur, you might ask? And how does a so-called resting neuron have a -70 millivolt electrical charge? Well, there are 2 factors that contribute to this negative resting membrane potential. First, the selective permeability of the membrane, and second, the uneven distribution of ions inside the cell compared to outside the cell. Now, let's get a visual of this process to get a better picture in our minds of what is happening at this microscopic level. Remember, we discussed in the last podcast that the cell membrane of all cells contain channels embedded in their phospholipid bilayer. These channels allow different particles and ions into and out of the cell. You can see 5 of them here. Now, what do you notice in this picture? There seems to be 3 different kinds of channels. One type, in purple, allows potassium to move freely into and out of the cell. These potassium channels are considered to be non-gated, just like a gate on a fence. The fence for potassium does not have a gate, and potassium is thus able to move freely from one side to the other without the use of any energy from the cell. It's important to note that only potassium can move into and out of this channel. No other particles or ions can cross at this channel. The second type of channel, in blue, seems to be a channel that allows sodium to cross the cell membrane. Except in this picture, it seems to be closed and deflecting sodium away, keeping it outside of the cell. Well, sodium channels in neurons are considered to be voltage gated, meaning that they will open and allow sodium through only if a certain voltage level is reached. And that will be the topic of our next week's podcast. But let's continue with our channels. The third channel on the far right is called the sodium-potassium pump. This channel does require energy in the form of ATP, and this energy is used to pump sodium and potassium at a rate of 3 sodium out of the cell for every 2 potassium into the cell. And we'll get back to this pump in a moment because it plays an important role in maintaining the resting membrane potential. So, this is why we say the cell membrane is selectively permeable, because it only allows certain ions and particles into and out of the cell, and sometimes only under certain circumstances, like the voltage-gated channel. Now, what is the consequence of this selectively permeable membrane? Take a look at the table above and note the concentration of these particles both inside and outside the cell. The most important element in what maintains the resting membrane potential are the numerous large proteins inside the cell that carry a large negative charge. This creates an overwhelming negative charge inside the cell compared to outside. These large proteins also affect the movement of potassium into and out of the cell. Because potassium is positively charged and can move freely into and out of the cell, thousands of potassium particles are drawn into the cell, attracted by the negative charge of the proteins. Remember, opposites attract. So, as potassium is drawn into the cell by this electrostatic gradient, the concentration gradient of potassium begins to build up inside the cell. Eventually, this concentration reaches a limit, and potassium is pushed out of the cell from the force of its concentration gradient. When the two forces on potassium are balanced, the membrane potential is still more negative inside. This is called the equilibrium potential for potassium, and it is reached at negative 70 millivolts, which is the resting membrane potential. As for sodium, because its channel is gated and sodium cannot enter the cell, sodium is in much greater concentration outside the cell compared to inside. This creates a positive charge outside. Furthermore, the sodium-potassium pump contributes to the resting membrane potential by pumping 3 sodium ions out of the cell for every 2 potassium ions pumped into the cell, keeping the inside of the cell negative. Therefore, the selective permeability of these 3 channels, potassium, sodium, and the pump, and their unique characteristics, generates a positive charge outside the cell and a negative charge inside the cell. This negative charge, again, occurs at negative 70 millivolts, and thus we say the cell is polarized in its resting state. This resting state of negative 70 millivolts is called the resting membrane potential. This means that the neuron is kind of on its toes, ready to respond to any incoming signals. If enough incoming signals disrupt this resting membrane potential of minus 70 and drive the charge up to minus 50, an action potential is fired. And again, this will be the topic of next week's podcast. This slide basically reviews what we have just discussed. If you are confused or need to review before moving on, please pause the podcast now. If not, let's move along. Now, once again, let's visualize this dynamic process at the microscopic level by watching this short clip.
The cell membrane of a neuron encloses cytoplasm with various ions dissolved in it. The neuron itself is immersed in a salt solution, the extracellular fluid. The ions of the cytoplasm consist mainly of positively charged potassium ions and large negatively charged organic molecules, such as proteins. Outside the cell, the extracellular fluid contains mostly positively charged sodium ions and negatively charged chloride ions. Since charged particles cannot pass through the lipids that make up cell membranes, they must travel through channel proteins that extend through the membrane. In an unstimulated neuron, only potassium ions can cross the membrane, traveling through specific proteins called potassium channels. Although sodium channels are also present, in unstimulated neurons, they remain closed. Since only potassium ions can cross the membrane, and potassium ions are more concentrated inside the cell, they diffuse out of the cell, leaving the large negatively charged organic ions behind. As more and more positively charged potassium ions leave, the inside of the cell becomes increasingly negative. But since opposite charges attract, as potassium ions diffuse out, an electrical force develops that tends to pull them back inside. At some point, the diffusion of potassium ions out of the neuron due to concentration differences is balanced by the electrical attraction tending to pull them back inside. This is the point at which neurons reach resting potential. Reaching resting potential in this way does not require significant changes in the potassium concentration inside or outside the cell. Only about 1/10,000th of the potassium ions initially inside a neuron must leave to create a resting potential of -60 millivolts. But it is action potentials, not resting potentials, that carry information through a nervous system.
Okay, let's quickly summarize the important concepts of this lesson. First, remember, all neurons have a difference in electrical charge across the membrane. This is called the resting membrane potential. Second, this resting membrane potential is negative, and therefore we say that the neuron is polarized in its resting state. And finally, this polarized state, or negative resting membrane potential, occurs because of the selective permeability of the membrane and the resulting uneven distribution of ions across the cell membrane. Stay tuned for next week's podcast as we discuss how this resting membrane is disturbed to generate an action potential and what this action potential does. If you would like more information on this topic, please refer to our class textbook or these websites. Thank you for joining me, and I hope to see you next week.